Hi, my name is Meg Younger. I'm an assistant professor in the Department of Biology and Center for Systems Neuroscience. So really, I'm a neuroscientist, and I'm excited to be here participating in this. I'm here because I work on mosquitoes. So after studying sort of model organisms like Drosophila for many years, I started working on mosquitoes because they kill an enormous number of people, which many people here have already addressed. And the way mosquitoes actually find people to bite relies heavily on their olfactory system. So mosquitoes smell us out. They detect our body odor, which is a complex blend of hundreds of different chemicals that they vary between people. And we know very little about human body odor and how the mosquito olfactory system works to detect human odor. And you can imagine that if we did understand this, we could come up with better strategies to stop mosquitoes from finding and biting people as they continue, unfortunately, to spread throughout the world, something we've heard a bit about and are going to hear much more about today. So at the onset of this work, we really didn't know very much about the mosquito olfactory system, but we knew a lot about how olfaction works in other organisms, especially the well-studied, workhorses for so much of neuroscience and biology, the fruit fly and the house mouse. And in both of these cases, there are these very big families of receptors that detect different volatile odorant chemicals. And there's the same organization you see in both of these animals, where individual olfactory receptors are expressed in individual sensory neurons, and each neuron only expresses one receptor type. Different neurons house different receptor types. And these different neurons project to these regions of the antenna lobe or olfactory bulb called glomeruli, which are thought of as processing units. And then these send projections to the deeper brain. And this canonical organization, which was first discovered in these organisms and has been seen in many others, allows for the detection of different odorant cues to be kept in these separate input streams and for a combinatorial code to be read out, encoding odor. So what we've seen is actually mosquitoes break all of these rules. Unlike the canonical olfactory systems, they have many more receptors than glomeruli, so they can't have this one-to-one organization. They co-express receptors pretty broadly in their olfactory system. And so unlike the canonical model, mosquitoes seem to have a non-canonical olfactory system where they co-express multiple receptors for different odorants in the same neuron. And what we want to know now is what role does this co-expression play in olfaction? And ultimately, the goal is how can we use that information to stop this really unstoppable animal from finding and biting people. So maybe this olfactory system is set up in a way that makes it so unbreakable to humans, which is why we're still using DEET, which was developed by the military in 1944. So thank you.